because mTOR complex 1 is important for growth, that means new protein synthesis, new mitochondrial synthesis, you allow the cell to make new proteins, make new mitochondria after you have turned up autophagy when you inhibited mTOR complex 1. So you break down the bad stuff, make the new stuff.
Hey longevity enthusiasts, it's time to take it to the next level. I've been fine-tuning my longevity regimen for years, and I look better and feel better than I did a decade ago. In fact, my blood work is even better than it was back then. And it's all because of my data-driven regimen, and it's inspired me to create a course and community just for you. It's called the Longevity Roadmap, and I urge you to check it out. If you're tired of your belly fat, tired of being tired, or just want to optimize yourself for the next 50 years, visit longevityroadmap.com. That's longevityroadmap.com. Welcome back to the show, everyone. Today, my guest on Longevity Roadmap is Dr. Matt Kaeberlein. He is a professor of laboratory medicine and pathology at the University of Washington. He's widely recognized for his groundbreaking research on the biological mechanisms of aging. He's got over 250 scientific publications, has received numerous prestigious awards, including the Klemier Award for Gerontologic Society of America, the Nathan W. Schock Award for the National Institutes of Aging, on Aging, I should say. And he's also the founding director of the UW Healthy Aging and Longevity Research Institute and co-director of the Dog Aging Project, which explores ways to extend health span in both humans and companion animals. As the CEO of American Aging Association, Dr. Kaeberlein continues to lead efforts in advancing interventions to improve quality of life as we age. Thank you so much for being on our show today.
Sure, my pleasure.
You know, I was telling you offline, I've been wanting to talk to you for a long time because before I started podcasting, and of course, I started listening to a lot of podcasts, and I heard you on a lot of shows. I think, gosh, a lot of interesting things to say. So let's start kind of broadly, though. When people think of aging, they often think of wear and tear over time. Now we know that's not entirely true. I mean, it certainly is true to a certain extent. Tell us the most damning evidence that there is something more to this than at the cellular level that might be programmed.
Yeah. So first of all, I think it's useful to clarify what we mean by programmed. And I say that because within, the scientific and certainly the biologists out there, when we talk about programmed aging, there's a lot of confusion, right? So most people, when we say programmed, what biologists or evolutionary biologists in particular really mean is that aging is selected for by evolution. I don't think there's any real evidence to support that in humans, at least. There are certain species like salmon where there may be evolved mechanism of senescence, but I don't think there's any real evidence in people that aging is a naturally selected program, meaning that's why our genes or those variants were selected for. But that doesn't mean that aging is not influenced by genes. In fact, we know aging is. And I think that's probably what you mean when you say program. So it's just useful to have that clarifying terminology. So what I think we can definitely say is, like almost any complex trait or simple trait, aging, the rate at which individuals age, how they age, is determined both by genetics and environment, right? It's a combination of those things. We can talk later if you'd like. I actually have a little bit of different thinking now on how important genetics versus environment is, but both of those things go into it. And we know there are individual genes that, when mutated, can influence the rate of aging, how fast we age, how slowly we age. So it's not only a sort of passive accumulation of damage. The rate at which that damage accumulates, the rate at which cells and tissues lose function, which we know goes along with biological aging, that is under the influence of both genetic and environmental control. And we partially understand that. We now have these things called the hallmarks of aging. There used to be 9, now there are 12. Next year there might be 15. But these are sort of our current understanding of the processes that seem to be intimately involved in biological aging, in determining why certain people age more quickly or more slowly, or why certain animals age more quickly or more slowly than other animals.
Yeah, it's interesting that distinction that you made just about program stuff. And you were right. I wasn't intending to necessarily go in sort of a apoptosis direction, but it did remind me of an interview I did with guy named Greg Fahey. Do you know the Fahey
Yeah, I know
protocol?
Greg
So,
really well. Yeah, in fact, he was
and
just on our podcast. Yeah.
yeah, and no, Greg is very interesting. His reasoning for focusing on the thymus,
Right.
right? The thymus being ultimately essentially sort of a parallel to the ovaries and producing eggs, and at some point they just stop producing eggs, and there
Mhm.
is some sort of programmatic element of that, right?
Yeah, it's interesting. So that's a good example, and I would suggest I don't know that we actually know what the selective pressure there is, right? So presumably there is some reason why the thymus senesces or at least declines in function. The idea that our thymus completely stops functioning, a little bit of a mischaracterization, but rapidly declines during puberty, right? And then we have this baseline level. But it is still an open question, I think, whether that was selected for, or was that decline in function, did that happen in the absence of any reason to continue functioning at a high level? And going back to aging, it's maybe easier for people to think of it this way, right? You think about, like, what is the primary job of any organism from the perspective of evolution by natural selection? It's to pass the genes on to the next generation successfully, right? There's lots of things that have to go into that. But once you've done that, evolution doesn't really care. You're obsolete, right?
Yep.
And so
Yep.
I think and again, it's a little bit speculative. It's hard to prove these evolutionary arguments, but I think the best evidence supports the idea that aging is largely because of the absence of selection. We've done our job. There is no reason to continue to maintain our bodies, our soma, in a high functioning state once we passed our genes on to the next generation. Some people will argue in certain long-lived animals like people, there is this thing called kin selection where there's some selective pressure for, especially on the maternal side, continuing to care for children and maybe even grandchildren. But that's probably a very, very small selective pressure. And largely aging is because once you've had kids,
Yeah,
evolution doesn't really care about you
we don't
anymore.
need you anymore.
Yeah, exactly. Yeah. And again, the only reason I bring it up is because you hear people get in debates or arguments within the field sometimes about programmed aging. And usually it's just because they're using the word differently. So I do think it is
Yeah.
important for people to
Yeah.
kind of understand that.
Well, let's kind of drive into some of those things that certainly are in the literature, at least.
Sure.
On the show, we've talked about some of those potential key drivers. Like, this is funny because I heard you on a show where this became controversial, but cellular senescence, mitochondrial dysfunction, epigenetic drift, loss of proteostasis. Tell us about some of the, what is a framework that you use when you think of biological aging?
Yeah. So I have a few frameworks, but putting it in the context of the hallmarks, which is sort of where you started there. So first of all, I think sometimes these controversies, they feel controversial to people who are outside the field, but they're really relatively small controversies about, nomenclature and things like that, right? But if you look at the hallmarks, I don't think there's a lot of argument around whether those things exist, whether they contribute to aging. And you mentioned 4 of them. Like I said, there's 12. I could probably get 8 or 9 off the top of my head. But they include things like, senescence, which you mentioned, mitochondrial dysfunction, DNA damage, epigenetic dysregulation, loss of proteostasis, inflammation. We go down the list. These are just sort of cellular, molecular changes that are at least associated with aging across the animal kingdom. And there's reason to believe they contribute to the functional declines, diseases, pathologies that go along with aging. Those are great. They're incomplete. That's number one. There's a lot we don't understand. And they're connected. And I think that's really important for people to appreciate. So when I think about biological aging, I really think more conceptually about the network that is sort of connecting the hallmarks. And why do I believe there's a network? Well, we know there are certain genes or proteins that play a role in most or all of the hallmarks. Things like mTOR, the mechanistic target of rapamycin, or AMP kinase, or FOXO, right? These things, when you manipulate them experimentally, affect most or all of the hallmarks at the same time, telling me there's an underlying network that connects the hallmarks. That's really what we mean when we talk about biological aging. And we know if we tweak certain nodes in that network, I just mentioned 3 of them, we can actually slow the aging process or speed it up, right? And we affect all of the hallmarks at the same time. So that's where I think the action is, and understanding that network. So there's really 2 things I think that we really need to do in this field. One is understand that network in as much detail as we can and figure out what are the most effective ways to target that network to improve health and longevity in people and animals. The other thing is figure out what we don't know, because I think there's a lot more that we don't know than we do know. So I think
Yeah.
those are really the two big things that the field should be focusing on right now. And I think we are. I think there are lots of people thinking particularly about this network and spending a lot of time trying to flesh out the mechanistic details of that network.
Do you think there could be, or what the likelihood of there being sort of a master regulator of the network? Or could we be looking at some of the hallmarks themselves? Like, for example, I know your work focuses on mTOR. Could
Right.
one of those not necessarily just be part of the network, but the master of the network?
I am inclined to not believe that there's a master regulator in the sense of something that we can, tweak it to stop aging. I don't think there's any evidence to believe that yet. I'm not saying it's impossible, but I've seen nothing that makes me believe that. I would frame it more as there are certain nodes in the network, key connection points, that are more amenable to experimental manipulation to have a positive or negative impact. They're more important in some ways. mTOR fits that bucket. We know we can experimentally turn mTOR up, and that accelerates aging. We can turn it down, and that decelerates aging. And we can get pretty big effects on lifespan and health span when we do that. We only know about a small number of these things. I talked about 3 of the most important already, which makes me believe that at least with our current understanding of the network, that it's a relatively limited number of nodes that we have the ability to manipulate at this point, to have big effects on longevity. There are other less important players where you can get smaller effects. I think the other thing, though, maybe this is what you were alluding to, an alternative approach, and there are companies and research groups taking this approach, would be to say, okay, we know what the hallmarks are. Let's not worry about the network. That's too complicated. Let's just try to deal with the hallmarks directly, or a single hallmark. So mitochondrial dysfunction, great example. Can we develop strategies, maybe antioxidants, maybe peptides that go to the mitochondria? I don't know if you've heard of SS31. That's a peptide that goes to the mitochondria and appears to have some beneficial effects on mitochondrial function. Certain metabolites. Maybe we can deal with the hallmark directly to have positive effects. And that can work. And I think from a drug development perspective, that makes a lot of sense if you're targeting individual diseases that are primarily driven by that hallmark. I still believe that you're likely to get much bigger effects and more broad effects on aging generally by going after the network than going after individual hallmarks in isolation. It's kind of like the difference between trying to cure an individual disease after somebody's got it versus trying to keep people healthy by targeting the biology of aging to slow all diseases simultaneously.
Well, how do you go about, other than, looking at all the different potential targets and targeting them separately through pharmacological means or whatever, how do you go after the network instead of the nodes?
Right.
Or are you talking about bigger things like, obviously exercise, things like that that we know are good for us, that probably drive all these things in the right direction?
Yeah. I think both are true. So I think the idea that lifestyle factors are the most effective ways to improve health and longevity in people is precisely because those things target the biology of aging. That is true. That's not a particularly useful way to understand the network,
Right.
but it's really important if you want to be able to give people advice on how they can give themselves the best opportunity to live a long, healthy life. I think the way we go after the network, there's a couple of strategies. And again, both of them are important and valuable and being pushed forward. One is to really try to understand at great mechanistic detail what the interconnections are. How does mTOR talk to AMP kinase, talk to FOXO, talk to the other players we know about in the network at a molecular level? Because as we understand those connections, it gives us more opportunities to both target that network more effectively and predict what might go wrong, right? Because sometimes we don't want some of these connections to be activated or inhibited. So that I think the field has made a lot of progress in. The other strategy, which unfortunately I think has been de-emphasized at the expense of the mechanistic studies, is sort of broad-based, unbiased discovery science. And that's how the network was originally found, right? Was instead of saying, we think mTOR is super important, so we're going to study it, this is kind of how I, got into the field. We did these unbiased genetic screens and let the biology tell us what's important. In other words, if you could look at every possible mutant or every possible drug, we can't do that obviously, but conceptually, if you could do that and ask, how do they all affect lifespan? Lifespan is the gold standard. And I'll tell you why lifespan is the gold standard, because the only way to robustly increase population lifespan is by targeting the biology of aging. Curing one disease will not have a large impact on population lifespan in the positive direction. It's really easy to shorten lifespan. Everybody knows a lot of ways they could shorten their own lifespan. But if you really want to increase population lifespan, you have to target the biology of aging. So let's look at 100,000, a million, 10 million different drugs and ask which ones increase lifespan by the most. Once we find them, that's valuable for two reasons. One, we've got drugs that increase lifespan by a lot, super cool. Two, we can use that to then ask, well, how are these drugs increasing lifespan? And that'll take us back. It'll either take us back to the network we understand and tell us something new about that network, or it will tell us about something we don't understand yet and tell us something new about the biology of aging. So I would really like to see more people doing that sort of unbiased discovery science, because I think we still have a lot to learn. And it's really, in my view, the best way to find things that are better than rapamycin or caloric restriction or metformin or whatever your favorite potential longevity drug is.
Well, since that's kind of where we're at, let's talk a little bit about mTOR, because I know that is something
Sure.
that you know a lot about, and I think it's a worthwhile thing for people to know about. But if you could just describe in your own words, what exactly is mTOR? What's the big deal about this? For people in longevity science, researchers like you, I think this is a special protein, and maybe you can talk a little bit about why.
Sure. Yeah. So like you said, mTOR is a protein. It's also a gene, but let's just focus on the protein. It stands for mechanistic target of rapamycin. It was actually named after the drug rapamycin, which is a drug that was found from soil samples on Easter Island, Rapa Nui. That's where the drug gets its name from. And mTOR was named after rapamycin because people discovered what rapamycin was doing was it's inhibiting or turning down mTOR. So why is mTOR important? Well, it turns out that mTOR is in many ways one of the central gatekeepers for a sort of fundamental decision that every cell that has ever existed has to make. Every cell, every organism that has ever existed has to sense the environment and make the sort of evolutionary decision, is it a good time to grow and reproduce or not, based on the environment? And if you think about what is it in the environment that helps inform that decision, there's only a few things, probably temperature, oxygen, maybe how much water is around, and food. And food is super important because if there's not very much food around and you have babies, from an evolutionary perspective, you're in trouble because you don't have anything to feed those babies. So you could think of mTOR as this gatekeeper. It senses all of those things, but primarily nutrients. And when there's lots of food around, mTOR gets turned up. That's a signal that tells the cell grow rapidly and at the organismal level, reproduce rapidly. When there's not much food around, mTOR gets turned down. That shuts off growth and promotes a more stress-resistant, resilient phenotype. And the speculation is, and I think it's reasonable speculation, is this allows the cell or the organism to survive the period where there's not much food around so that when the environment changes, you can grow rapidly and reproduce. So that's what mTOR does. Now, why is it important? Well, if we go back to the sort of discovery science, there were 4 or 5 labs, and I was fortunate to be involved in some of this research. This was in the early 2000s, so 2003, 2004, 4 or 5 labs independently stumbled on mTOR as an important regulator of longevity. And I think that independent part is really important because we were not going into it thinking this was going to be important. Each of us, for our own reasons, hit upon mTOR and found when we turned it down in budding yeast, in nematode worms, and fruit flies, organisms that are billions of years separated by evolution, in all of those places, it increased lifespan. And then a few years later in mice, the interventions testing program from the National Institute on Aging found that rapamycin given to mice also increased lifespan. So suddenly we had this pathway, this protein, this drug that in every organism, every major model organism, if you gave it to that organism, would slow aging, increase lifespan. And we've learned in the last, this was 2009 when the first study in mice was published. So in the last 16 years, that not only does it increase lifespan in mice, it does it reproducibly. It works for everybody, which not everything in this field does. Up to about a 30% increase in lifespan. So pretty big effect in mice. You can start the treatment in middle age, which is really important. We can talk about why, but that's really important if you want to think about use in humans or companion animals at some point. And it seems to delay, if not almost every aspect of aging and every tissue and organ where it's been tested. And in some places, you can even start the treatment in animals where there's been a functional decline. So, for example, you can see in the heart, when you look in mice, there's an age-related decline in function, in the ability of the heart to contract and pump blood. You start giving rapamycin to the mouse equivalent of a 60-year-old person, that function gets better. So that's true in the heart, it's true in the brain, it's true in the immune system, it's true in the oral cavity, and it's true in the ovaries. You can actually restore lost function in an aged animal. So again, really exciting from a potential translational perspective if we can show, and this hasn't been shown yet, but if we can show that the same thing's true in people or potentially in our companion animals.
So what is the highest level organism that's really been established in?
Yeah. So for the functional improvements in health span, mice and rats, both, for lifespan, I'm not sure if it's published yet, but there's been data that's been presented at the American Aging Association meeting in marmosets, which is a non-human primate, that rapamycin can increase lifespan. And then in dogs, we found at least early evidence for improvements in heart function and activity with short-term treatment. So it's starting to make a pretty compelling case. This is not going to be only in rodents, only in the laboratory, that is probably going to be true in primates, at least, non-human primates, and for at least some of the functional measures, potentially in dogs living in the human environment.
When you're inhibiting mTOR, obviously you're turning off growth, but what does that mean then? Is it autophagy? Yeah. Are we talking about, so is that the primary thing that's happening?
I mean, it's one of the things. So I think there's a few things to say here, right? It's all about dose, right? So you don't want to, with any of these things, you don't want to go too far in one direction or the other. So in many ways, rapamycin is what people think of as what's called a caloric restriction mimetic. So I talked about how mTOR senses nutrients. Calorie restriction is the most reproducible and robust intervention for increasing lifespan in rodents. I don't recommend that level of caloric restriction in people, and we can talk about why I think rodents and people are different there. So you can think of caloric restriction as like a sledgehammer, right? Rapamycin is more like a precision tool where caloric restriction is going to turn down mTOR and it's going to do about 50,000 other things. Rapamycin goes in and specifically turns down mTOR. So why is that beneficial? I think turning up autophagy is one of the likely mechanisms, and there's certainly preclinical data in simple invertebrate models to support that. By the way, this whole idea that fasting works because it turns up autophagy, first of all, too simplistic, but it is an overlap with rapamycin. The reason fasting turns up autophagy is because fasting turns down mTOR, which is what rapamycin does.
Yeah.
So that's certainly part of the story. I think in mammals and, in the people anecdotally where I have observed potential benefits, there's also this very potent anti-inflammatory effect. And we haven't talked about rapamycin in the clinical setting. Rapamycin is FDA approved. It was first approved for use in organ transplant patients. It's going to be a different dosing schedule than what we're talking about for longevity. But in that context, rapamycin has the ability to blunt the immune system. That's how it protects organ transplant patients from organ rejection. And so it seems to be very potent as an anti-inflammatory at lower doses, particularly towards the types of inflammation that are associated with aging. And sort of as a class, this is an oversimplification, but immunology is really freaking complicated. And so I don't want to get into the weeds there because I don't understand it. As an oversimplification, right, with aging, we have an increase in what people call sterile inflammation, right? Meaning it's the immune system often responding to things that when you're young, typically wouldn't respond to, right? Autoimmunity is one class of this. And rapamycin seems very potent at blunting that sort of sterile inflammation. And we know chronic inflammation is one of the 12 hallmarks of aging, and we know inflammation is associated with a bunch of age-related diseases. So I think autophagy is important. I think the blunting of chronic inflammation is important. My lab was really the first to show that rapamycin could suppress mitochondrial dysfunction. We did this in a mouse model of a severe childhood mitochondrial disease, but now we know there's all sorts of mitochondrial dysfunction that rapamycin can improve independent, apparently, of autophagy. So there's probably more than one thing that, and this is sort of what I was alluding to earlier. If you turn down mTOR, you actually affect all of the hallmarks of aging, some of them more than others, but this is that interconnection of that network where it really seems like rapamycin is really slowing the biological aging process writ large, as opposed to only affecting one hallmark.
So there is From the clinical side, people who are taking it are pulsing it typically, anywhere
In
from,
off-label use. Yeah, that's right.
off-label use, maybe
Yeah,
6 to 10 milligrams once a week. But could you explain the concept behind the pulsing? Because I
sure.
believe there's a mTOR1, mTOR2 nuance there that might be useful for people.
Sure. Yeah. So let me start by saying sort of my understanding of how that evolved, and then we can get into the speculative mechanism. I just think I'll talk about the mechanism. I think it's
Yeah,
important for people to understand it's still kind of speculative as to whether
right.
that's really what's going on. So the reason that pulsing evolved, I believe, really stemmed from two factors. One was a series of studies that Joan Manick did. I think she probably started in like 2009, where she was at Novartis at the time and then started a company called Restore Bio. And they were interested in the idea that short-term treatment with rapamycin or a derivative of rapamycin called everolimus, you can just think of them as
Uh-huh.
exactly the same
Yeah.
molecule, same biochemical activity, whether short-term treatment with everolimus could boost immune function. And that was based right off of work in mice that Pan Zhang's lab did, showing that's exactly what happens in H mice. So they did a series of clinical trials in healthy older people. These were people between 65 and 85, and they treated them for 6 weeks with everolimus. And in the first trial that was published, I think it was published in 2011, 2012, they tested 3 doses. They tested a daily dose. It was a few mgs a day. They tested weekly 20 mgs once a week, and they tested weekly 5 mgs once a week. And what they found was all 3 doses
Good.
had efficacy in terms of it appeared to boost the vaccine response to influenza. So these people got a vaccine after their 6 weeks. The 20 and 5 mg compared to the daily, if I recall correctly, had better efficacy and also better side effect profiles. And at 5 mgs once a week, the side effects were no different than placebo. So that was one piece of evidence that this once weekly dosing could be effective and reduce side effects. And then there was a doctor named Alan Green who was sort of ahead of his time, who because he had hypertrophic cardiomyopathy, started taking rapamycin for himself. It seemed to at least stop progression of HCM, if not actually partially reverse it in him. And he started prescribing it off label. And he was really the first to do this at scale. And he settled on 6 milligrams once a week. And he had his own rationale for it. I'm not criticizing it at all. I think it's a reasonable rationale. So a lot of people started taking it at that dosing schedule. So there's this cultural, and I mean, this is sort of the nature with off-label use. Doctors talk to each other, patients talk to each other. People are like, okay, this seems to work, so we'll stick with it. So that's how it's evolved today. And then in parallel, a potential mechanistic explanation for this reduction in side effects has emerged related to the 2 complexes that mTOR acts in. So mTOR protein, there's one protein, mTOR. That protein can function in at least 2 distinct complexes with other proteins. mTOR complex 1 and mTOR complex 2, or you'll hear them referred to as mTORC1, mTORC2. Okay. So, and the complexes do different things. So mTORC1 primarily involved in nutrient sensing, regulating growth, the stuff I was talking about before as being important for aging. mTORC2, less studied, but it seems to interact in interesting ways with AKT and PI3 kinase signaling, regulates actin cytoskeleton, probably does a bunch of other important stuff. And it's useful for people to understand both complexes are essential. What I mean by that is if you're null, if you have 0 activity in either mTORC1 or mTORC2 at the organismal level or the single cell level, you are dead. They are necessary for life, absolutely essential. Rapamycin is a specific inhibitor of mTOR complex 1, and it does that through what's referred to as an allosteric mechanism. This is getting in the weeds a little bit,
Yeah.
but
No, it's
it's
good.
probably a little bit relevant as well, because there are other classes of mTOR inhibitors that act through a different mechanism. So rapamycin is an allosteric inhibitor of mTOR complex 1. The way it works is it actually gets into the cell and binds a protein called FKBP12. Once it's bound to FKBP12, that complex then goes and disrupts mTOR complex 1, doesn't touch mTOR complex 2. So rapamycin is a specific inhibitor of mTOR complex 1. Great. We know genetically turning down mTOR complex 1 slows aging, increases lifespan, mimics calorie restriction, all the good stuff. That's the
Yep.
simplistic way of putting it. It's not
Yep.
accurate, but it's
Sure.
the simplistic way of
Yeah,
putting
yeah.
it. Okay, so turning down mTOR complex 1, good. It turns out, though, if you have mTOR complex 1 too low for too long, there's this feedback mechanism that then impacts mTOR complex 2 and actually degrades components of that complex. So the current thinking is, and there's a little bit of evidence to support it, it's that chronic inhibition of mTOR complex 1. So daily dosing of rapamycin,
Right.
for example, would lead to chronic inhibition of mTOR complex 1 that then has a feedback indirect mechanism on mTOR complex 2, and the side effects come from that feedback on mTOR complex 2. Okay, so I am not convinced, but it's a plausible explanation that that's what's happening, and that all the side effects are coming from inhibition of mTOR complex 2. So the idea is if you cycle rapamycin, you'll get acutely high levels of rapamycin, which will strongly inhibit mTOR complex 1, but within a few days, you'll go back down to no rapamycin in the system. You return mTOR complex 1 levels to its normal state, and you get a break before the next dose of rapamycin. So you don't get these off-target effects on mTOR complex 2. The other thing you get, because mTOR complex 1 is important for growth, that means new protein synthesis, new mitochondrial synthesis, you allow the cell to make new proteins, make new mitochondria after you have turned up autophagy when you inhibited mTOR complex 1. So you break down the bad stuff, make the new stuff.
It all sounds like it makes a lot of sense, but then when the one thing that I always think about is, well, you go back to the ITP, right? Interventionist testing program with the rodents, they were not pulsed, right? They were just
daily. Yeah. In fact, it's more than it would be even in people daily taking a pill, because it was in their food in an enteric-coated, encapsulated form. So the mice are going to eat throughout the entire night. So it's almost like you're doing like an IV drip of rapamycin 12 hours a day or something like that.
Yeah. So that obviously makes it a little tricky, because then you're like, well, I mean, what was the effect of that? And then, of course, rodents only die of cancer anyway, so that's
not true. No, wait a minute, that's a myth.
That's not true. That's what I thought.
So,
isn't that the most common cause, though?
So that is true. Well, so the most common this is getting a little pedantic, but the most common cause of death in rodents is actually euthanasia, because we're required to euthanize them.
Right.
but certainly, here's the way I would frame it. All mice in the laboratory, naturally, like wild type mice,
right?
There are all sorts of weird genetic models, but
okay.
Wild type mice, which is what we're interested in if we're interested in aging, die with cancer. How many of them are actually euthanized because of cancer depends a little bit on the strain background, but it's nowhere near 100%. They get a bunch of other age-related diseases, just like people do. Actually, a lot of them end up having to be euthanized because of skin diseases that are age-related. So there's a bunch of kidney disease as well. Some mouse strains get.
Well, I mean, that's really good to know, because, like, I think the popular, at least clinically, like guys like me who are just follow the science but are not scientists, are in it. I mean, I think we're all kind of under the impression that, well, these rodents are all dying of cancer anyway. So what does that tell us? Right?
I think it's a really important. I think that's exactly right, because there's this misperception. You'll hear people say this sometimes, like, "Oh, caloric restriction only increases lifespan in mice because it is anti-cancer."
Right.
And first of all, that's not true. That's the only reason it increases lifespan in mice, but it misses the point. There's this tome. I mean, this is an amazing tome by Roy Walford and Rick Weindruck I think it's called The Retardation of Aging by Caloric Restriction. It was written like in the 1990s. They've got this book of all the tissues and organs and pathologies and diseases that caloric restriction stops. It stops everything that goes along with aging, pretty much. So it's anti-cancer and everything else that goes along with aging. And yet these, this simplistic idea that it's all cancer somehow gets out into the mainstream sort of mindset, and it's just wrong. And you hear that with rapamycin too. So it is important for people to understand, even if it's a potent anti-cancer mechanism, and it's still slowing aging in all of these other tissues and organs and preventing the functional declines. And at least with rapamycin, at least partially reversing some of these functional declines if you start treatment in middle age.
Let me ask you this, man, and from the clinical side, this is something that's always kind of vexed me. When you're working out and you're building a lot of muscle, you're cranking up mTOR.
Right.
And we know that lean muscle mass is one of the most important variables, if not the most important variable, for longevity in middle-aged, humans.
Yep.
So
certainly health span, at least.
Yeah, in health span. So how should we see that dichotomy?
I think that's a great point. I mean, the honest answer is I don't think anybody, the muscle biologist, the rapamycin biologist, I don't think any of us really understand the intricate inner workings of mTOR and muscle formation, muscle maintenance in people in the context of aging. So I think we have to be guided by the actual data. right? So here's the data that I'm aware of. So people thought rodents taking rapamycin would lose muscle and develop what's called sarcopenia or
Right.
frailty. So sarcopenia is loss of muscle mass and invasion of fat into muscle. Frailty is what it sounds like, frailty. So that's what they thought. And that's what all the muscle biologists were saying. Rapamycin is terrible. It's going to cause these mice to get frail and die. And then somebody did the experiment, and by God, they live longer and they have more muscle later in life. And the muscle looks better by, all sorts of metrics. So in rodents, rapamycin preserves muscle mass and muscle function in the context of aging. That is clear. No question about it. So then the muscle biologists were like, well, those are just mice. It'll be in people, it's going to be terrible. Everybody's going to get weak and get frail. The honest answer is we don't have the double-blind controlled clinical trials yet. The data that's out there, and there was actually, it's not perfect, but it's okay. There was this Pearl trial, which was a distributed clinical trial. It was placebo-controlled. I mean, people using rapamycin off-label, very low dose. I think the people ended up getting about 2 milligrams a week, so lower than even what we were talking about. And at least in women, it seemed to preserve muscle mass. And there's a bunch of anecdotal reports that I'm aware of where you actually see positive changes in body composition in people using rapamycin off-label, either in combination or even a little bit without dedicated resistance training. So how do we explain that? Given that we know turning up mTOR is necessary for building new muscle. So first of all, I would say we need to look at the data first, and then we can develop hypotheses to explain the data. But the data are what they are. So if it is the case that rapamycin preserves muscle mass and improves body composition, that's important, even if we don't understand why. Okay, so we don't know that 100%, but I'd say that's where the data is leaning at this point. There are other clinical trials happening. Brad Stanfield has one out of New Zealand. I haven't seen the data yet, but it's my understanding they have completed collecting the data. So we'll see there, looking at muscle function in elderly people with rapamycin.
No, I should point out, I did interview one, I wish I remembered who it was, but a really good scientist who related to exercise, who had some data, and I don't remember if it was human data, I don't remember what it was, but showing that the relative increase in activation of mTOR was more during exercise, and weightlifting was more isolated to muscle tissue.
Yes.
Which would make a lot of sense, right?
Absolutely.
Right, because then you're not getting a systemic increase in mTOR, which could be problematic for all the other reasons we've talked about, but you're building muscle.
Right. So I think that's a good point. So this is now, how do we explain the observation? If it is the case that rapamycin is actually beneficial for muscle in aging, at least at off-label doses, how do we explain that? And one is what you just suggested, which is that mTOR activation to build muscle is going to be localized to muscle. How much of the rapamycin at off-label doses is getting to muscle and inhibiting mTOR is a little bit unclear. And it's going to depend on the dose. But remember, we're not, in most people, doing this every day. We're doing it once a week. So you're going to get this acute inhibition of mTOR, even if it's in muscle, for one day of the week. And then the other 6 days, you can go ahead and have normal mTOR function. I think, how do you explain better outcomes with rapamycin? I think the anti-inflammatory effect is potentially part of it. I think the improved mitochondrial function is potentially part of it. And then I think, we haven't talked about it, there's a lot of data in the preclinical literature on how rapamycin can affect the distribution of fat burning versus burning sugars and carbohydrates. It may also be that rapamycin is promoting a shift towards burning fat, and that also contributes to the positive changes in body composition. But it's very speculative.
Yeah.
Again, I think the first principle is, let's see what the data show us. And then if we see evidence for improved body composition, then we can go back and try to figure out how. I think what I feel pretty confident with is that many people, I don't want to say everyone because nothing is all or nothing, but most people, I would say, can build muscle and maintain muscle using off-label doses of rapamycin.
Yeah, I want to make sure we cover some other things because I've just got so many questions. But, with the whole longevity space was really kind of it took off with David Sinclair's book, right? And Sinclair really, I think, got in my head too, like this whole idea that really we've kind of solved this concept by saying that all that aging is loss of information. It is epigenetic drift.
Right.
And obviously the more I talk to people like you and others, that's obviously not necessarily what everybody believes, that this is probably, what I'm gathering from you is that this is one of the hallmarks of aging. It's one of the nodes. It's not the cause.
Yep.
Knowing that, what do you make of, because a lot of people are talking about epigenetic clocks that are based essentially on epigenetic drift, and can you give me your take on that and your thoughts?
Sure. Yeah. So there's a lot to unpack there. First, I will say I agree with you. I mean, I think the reason why the longevity field has taken off is there's a variety of factors that fit into that. But absolutely, David is a fantastic communicator and has had an impact on popularization of the topic. He also sometimes oversimplifies and says things that aren't true, I'll just say.
Like
And
like
then sometimes he
resveratrol,
exaggerates.
like the use of
So anyways,
resveratrol.
yeah, we don't need to talk about resveratrol. The only thing I'll say about resveratrol, it is the
Yeah.
most debunked anti-aging drug ever. Disproven
Well, that's
over and
good.
over and over
I'm glad
again,
you said that because
meta-analysis.
you need to knock that one out.
It's again, I love Mark
Yeah.
Hyman. I agree with
Yeah.
95% of what he says. I was at a conference and it was great until he started talking about sirtuins and resveratrol. I was like, oh my
Yeah.
God.
Yeah.
Anyways, that's a different topic.
Yeah.
Okay.
Yeah.
So
Yeah.
epigenetics. So first thing I'll say is they are one of the 12 hallmarks. Now, does that mean they're only 1/12 of aging? I don't know. Nobody knows. So could they be the most important hallmark? Absolutely. Could they be just one of the 12? We don't know is the real answer. David certainly believes they are more important than the other hallmarks. He's entitled
Yeah.
to his opinion. I just wish he wouldn't say his opinion was fact because it's not, because it's yet to be established. Why would people think, though, that epigenetic changes are more important? And I think there are reasons to believe this. So first of all, definitionally, what do I mean? Okay, so epigenetic changes are modifications on top of the genome, right? Our genome is our genetic code. That's what you get sequenced, GATC, right? All of that. DNA damage is one of the hallmarks of aging. Those are mutations to your genome that happen with age. We know lots of mutations cause cancer, for example. Epigenetic changes are on top of that. So even if we could completely fix the epigenetic changes, you're still going to have DNA damage. I am rock solid certain epigenetic changes are not 100% of aging because you're not going to fix those mutations, period, full stop. Okay, that's clear. So why might epigenetic changes be really important? Because these modifications on top of the DNA, and when people are talking about epigenetic clocks, they're mostly talking about DNA methylation, but there are a whole bunch of these epigenetic modifications which have yet to be fully cataloged. There are modifications happening that people can see by mass spec that we haven't documented or characterized yet. So, but DNA methylation is one of these important epigenetic modifications. And what happens is by methylating certain positions in the genome, you change which genes get turned up and which genes get turned down. And of course, it's the expression of the genes that leads to more or less of that protein, which then leads to all of the different ways that different cells function, including differentiation. So epigenetic changes are how we get from a pluripotent cell that can become any cell type, a neuron, a fibroblast, a cardiomyocyte, pick your favorite cell type. We have pluripotent cells that can become any of those things, and they become differentiated or specialized by specific epigenetic changes that control which genes get turned up and which genes get turned down. People may have heard of the Yamanaka factors. Those are factors you can put into differentiated cells, fibroblasts, neurons, cardiomyocytes, revert their epigenome back to the pluripotent state, and then they become pluripotent again. So super powerful technology. So with aging, we know there are other changes. It's not the changes that happen to take a pluripotent cell to a fibroblast, but there are other changes that happen in a characteristic way with age. And what these clocks do is they measure a subset of those changes, usually on the order of a few dozen, maybe 100 specific DNA methylation marks that change with age. And because it's characteristic, we can determine with some level of precision your chronological age, how many birthdays you've had based on your methylation pattern in those specific marks. A different set of marks are correlated with mortality, your risk of dying. People call that biological age. It's mortality and biological age are related, but they're not perfectly correlated. So you can find epigenetic marks that correlate with your risk of dying in 3 years, 5 years, or 10 years. That's what people are calling biological age metrics. Okay. So people have developed these clocks. They work really well in the research world. And then people are selling them in the direct to consumer space. I'll just say three things about the direct to consumer clocks. One is, I already said it, it's a correlation to a correlation. So it's not actually measuring biological age. It's just a fact. They'll call it biological age. It's not. They don't work. And I've shown this. Check out the Optisan podcast if you want. But I did an experiment where I took 8 of them. So 4 different tests by 4 different companies, did it in technical replicates, same biological sample, same day. I was 53 3/4 on the day I did it. I got results from 42 to 64, right? All over the map.
Yeah.
They are not accurate.
They're not accurate or precise, basically.
That's right. Yeah, I guess I shouldn't say they're not accurate because I don't know what my true biological age is because we don't have a way to measure it. You're absolutely right. They're not precise or accurate because it's all over the place. And the third thing that I think people don't realize is none of these tests have been validated. None of them are FDA approved. None of them have been vetted by an independent body. These companies just say, we're measuring biological age, pay $500 for our test and we'll tell you a number, right?
So when you tested it, none of them were precise either. In other words, none of them gave you the same number over and over? Because
good question.
I can see a situation where you might just want to have a relative sort of idea that, you're getting better or worse or something like that.
So what I only did 2 replicates of each one. So I don't have enough to really know precision beyond, just those 2. 3 of the companies were relatively close together within maybe 4 years of each other, the 2 values. One of them was 20 years apart. So this is, I think, the interesting comment that I get a lot, which is that, okay, well, maybe none of the tests are very good, but if you had longitudinal data from the same company, right, the same test over many years, it would be that change, that delta over time that matters. And my response to that is, yeah, maybe, but we have no reason to believe that, right? So
even if the best study, if the best one you had was 4 years, that's 4 years is a long time.
It is. Yeah. Again, this is part of the problem is I don't want to say, I mean, part of me does want to say it just to be provocative, but I can't say that none of these tests work, right? Maybe one of the company's tests was spot on. The problem is we have absolutely no way to know because there's no transparency and the companies aren't providing any transparency into what the variance is. If you take the same test 18 times on the same day, what does that variance look like? How well is this correlated to anything we really care about? We just don't know. The other piece is they're not really actionable right now. And maybe it's worth also mentioning there's this thing called pace of aging, right? So that's another way that the people who want to sell these tests are like, okay, well, it doesn't really matter if the clocks are accurate or not, because what we're really interested in is how fast you're aging. And we have this different test that will measure your pace of aging. It's based off of a study called the Dunedin Pace of Aging study, right? Where they actually found a different set of methylation marks that were correlated with how fast people's health trajectories were changing over a 10 or 20-year period. Again, super cool from a research perspective. We have no way to understand whether what these companies are actually reporting as the pace of aging makes any sense or is biologically accurate or precise. What I can tell you is I have data from the same company that reports their biological age metric and the pace of aging over a 2.5-year period now. My biological age was going up and down by, a decade over this 2-year period. My pace of aging the entire time was less than 1, meaning I should have been aging more slowly biologically than the average person. And yet my absolute number was going up and down. So all I can say is within the same company, the pace of aging wasn't matching their biological age metric. So I see no reason to trust these companies right now. Like, why would you trust somebody who says, I will sell you a test that tells you X with no evidence that it's actually working?
Yeah. Well, maybe something it'll get better. Who knows? But
I think it will. No, and again, this is where I come off as very negative on the biological age clocks. I think from a research perspective, they are really, really powerful and getting better. They'll be really valuable at some point, even potentially for clinical trials, right? If you could use these biological age tests or the pace of aging algorithm to predict whether a drug is working or not early on, that's super valuable. I think the problem is this rush to market, right? And the lack of any, the incentive structures are just misaligned, right? There's no regulation, so there's no incentive to do it right. The incentive is to sell stuff to people. You don't even really care whether it works. I think some of these people are well-intentioned. I think they do care, but the incentives are always going to be to rush it to market to make money and to shortchange the quality control. That's the real problem, the way I see it, is in the industry.
Yeah. When we talk about some of the other, I just want to run through a few. I know it's hard to get into any of these without a significant discussion, but
I know I have a hard time with that. Yeah.
I know. Well, no, and it's great. This is what I want, but I also know you have a other things to do today. Really. So, but I do want to touch on a few things. For example, I mean, what's your take? What's your view? I'll tell you, when we talk about metformin, right?
Mhm.
Metformin, I personally, when I just look at the literature, I feel like I'm pretty convinced that that metformin's real value is in euglycemia,
Mhm.
is in probably the anti-inflammatory effects of euglycemia, and that it's not some, special geroprotective drug. There was the initial study, I forget the name of the author on that suggested that there was, people on metformin
Right.
who were not diabetics were outliving, or people who were diabetics on metformin were outliving their counterparts who were not diabetics. It sounded
Right.
fantastic, but that study was full of flaws. Tell
Right.
me what your take on metformin is, because
Yeah.
obviously this is another one where, in the longevity space, it's like, yeah, everybody's on metformin.
Right. Yeah. So my take has been consistent for the last 5 years.
Yeah.
And it very much aligns, it sounds like, with where you're at. So first of all, I think it's useful. So when I think of a longevity drug, I start from first principles, and really ask, do we have evidence that this increases lifespan? Health span is super important,
Yeah.
but from evaluating, is this really targeting the biology of aging? This goes back to what I was saying before. The best way to know it targets the biology of aging is if it increases population lifespan by 20, 25%. Then you can be rock solid sure. So you go back to the mouse data, because obviously we don't have that yet in people. Go back to the mouse data. The studies are pretty clear. In healthy, normal, wild-type mice, metformin has no effect on lifespan unless you give too much, and then it shortens lifespan. There's this really funny study by Rafa DeCabo at NIA where they tested 2 doses of metformin. I don't remember the specific doses, but they tested 2 doses, low and high. Let's just call it that. So the low dose increased lifespan by like 3%. Tiny effect, right? If
Yeah.
anything, probably not even real. The high dose shortened lifespan by like 10%. And yet the title of the paper was Metformin Increases Lifespan and Health Span in Mice. Talk about selective interpretation of your data.
Yeah. Right.
And then
right.
the ITP, the gold standard, tested metformin, had no effect. So there's really no good reason from preclinical studies, animal studies, to believe metformin is a potent modulator of this network. Having said that, metformin does activate AMP kinase, at least in cells. AMP kinase is one of the nodes in the network. So you can make a hand-wavy, plausible argument. But here's where it gets important. How does metformin activate AMP kinase? Nobody really knows. The best guess is it's an inhibitor of the electron transport chain, complex 1 specifically. We know that inhibiting complex 1 too much is a really, really bad idea. It causes
like
all sorts
cyanide.
of mitochondrial disease. Yeah, right. So you got to be
Right.
careful in that regard. Although metformin really is a pretty safe drug. I don't want to scare people. Okay, so what do we know in humans? Really good diabetes drug. Diabetics on metformin do great compared to diabetics that aren't taking anything or aren't taking metformin. The study you mentioned is interesting. A lot of people got excited because it suggested that diabetics taking metformin, at least some of them, were outliving non-diabetics not taking metformin. If that was real, that would be super exciting. Turns out it probably isn't real, because other studies now have come and failed to reproduce that, and many people have pointed to the methodological flaws in the first study. So what do we know about metformin beyond that? And this is where I think I have never been bullish on metformin as a longevity drug in people, because I think there are, even though the side effects are not terrible, especially if you're a diabetic and you need to be taking something, but there are some side effects people should be aware of, right? So I don't know what the percentage is. Some percentage of men taking metformin see a drop in testosterone. Okay, we can have a conversation about testosterone, but if muscle mass and bone density are important, and I think they are, you probably want to be aware if you have low testosterone, aside from all the other stuff that goes wrong with having low testosterone. Then the other piece is, and this is new, but there seem to be more studies supporting this, and now some evidence in people, that metformin may blunt the beneficial effects of exercise. That's probably because it's a mitochondrial inhibitor. Makes sense. So if you're exercising regularly, we should all be exercising regularly. You might want to consider, especially if you're not diabetic, whether you want to take metformin. And then the last piece I'll put on top of that is there are other anti-diabetes drugs that seem more promising from what we would call a gerotherapeutic or a longevity perspective. The ones I think that are really interesting are these SGLT2 inhibitors,
Right.
canagliflozin
No one
and
talks about them.
empagliflozin.
In
Yeah.
in clinicals, in the clinical world, no one talks about these in the longevity space, which is really
Yeah. So
interesting to me because
let me just
as
tell you the data,
yeah,
or
yeah.
maybe you know it, but
Well,
it's really
canagliflozin was one of the ones that actually did show benefit in the ITP, correct?
that's right.
That's right.
And that's right. So it came out of the interventionist testing program in mice as increasing lifespan. And there was a study that looked in UK Biobank at all prescription medications. And then they had to have a certain number of people who'd gotten it. And they compared it to people with similar health status who didn't get that drug. And there were like 12 drugs that came out associated with lower all-cause mortality. A bunch of them were estrogens and estradiols, super interesting, also came out of the ITP as increasing lifespan. And then the SGLT2 inhibitors came out. So
Right.
to me, it doesn't prove anything other
Right.
than there's an association with lower mortality in people. Oh, and these things seem to slow biological aging in mice. That's pretty intriguing. And they're effective anti-diabetes drugs. And it seems like there may be some kidney protective effects. So if I was going to take an anti-diabetes drug, not being diabetic, for longevity, I would lean towards SGLT2 inhibitors over metformin.
Got to ask you about NAD boosters in particular.
Mhm.
You know, I mean, I've had Charles Brenner on, who's pretty. He's very opinionated.
He's got strong opinions, super smart guy, fantastic biochemist, but very strong opinions.
Yeah. He cringes at the idea of longevity, right? The word longevity, he hates the length.
Yeah.
But he is all about, in particular, NR, which his brand is Niagen, the one that he's behind, does improve significantly life, or health span, I should say. So what's the study there in NAD boosters?
So again, much like metformin, the preclinical literature on NAD boosters is far from compelling, I would say. So again, early studies suggesting increases in lifespan, the ITP failed to replicate that. And this space, the NAD boosters in general, is full of preclinically studies that were not reproducible. So a lot of smoke. I don't know if there's any fire there preclinically, but what I would say is these don't rise to the top in terms of interventions that have a robust effect on lifespan or health span in mice. In people, also, I would say it's unclear. There have been a few clinical trials that are suggestive. They're all small. They're all underpowered. They're all done by people with a vested interest in seeing a benefit. But certainly, could there be benefits from NAD precursors in people? Absolutely. My guess is, and again, this is my personal opinion, sort of looking at all of the literature that I'm aware of. I don't think NAD levels generally robustly decline with aging in people in most tissues. That's sort of I don't want to say it's a myth, but it's a dogma that has been propagated by a small number of people with a vested interest in getting grants and making that seem like it's a fact. It hasn't been shown. It may be the case, though. I think it is the case in certain people with certain metabolic conditions that NAD homeostasis is perturbed. And I think NAD precursors may be beneficial in that context. Then we can get to which NAD precursor. There's at least 4 of them you can think about, and also IV NAD or sublingual NAD. So the 4 I think that most people will have thought about: nicotinamide riboside, you mentioned, NR, nicotinamide mononucleotide, NMN, that's David Sinclair's variant, nicotinamide, and niacin. So they all are NAD precursors. If you take them orally and you take enough of them, they will boost NAD levels in blood, at least until your liver clears it. And then you'll make a bunch of other metabolites. The only evidence I know arguing for maybe some caution with NAD precursors, and it's minimal, is there's one study in aged mice that suggests that at least high levels of NMN lead to high levels of liver metabolic byproduct called 1-methyl-nicotinamide, which can then go cause pathology in the aged kidney. So again, it's pretty speculative, one study, but for people who have kidney problems, or this is where I really have concerns, companion animals, dogs and cats, where there are lots of kidney problems, I would be cautious about giving an NAD precursor to an aged companion animal or to an aged person with kidney pathology. I've tried a few different NAD precursors. I've never felt anything. That means nothing.
Yeah.
So, take it for what it's worth.
Is there any supplements out there that do intrigue you? Like, for example, the other ones that comes to mind for me, just because one study I remember seeing on taurine.
Yeah.
Taurine being again, pretty, I mean, maybe you tell the story. It might make more sense to,
Sure. I
you
mean,
know,
taurine's an amino acid. It's involved in again, these things are all super complicated. So it's
yeah,
involved in a bunch of different biochemical and biosynthetic
yeah,
processes. I was actually a co-author on that study, a very minor role. But
yeah,
what that study showed was that in mice and in people, there's evidence for a decline in taurine with age. I would also put that in the category of NAD. Like, I don't want
yeah,
to say
right.
that's a general thing. I don't think it's the case that everybody's taurine levels decline with age, but across the population, there's a correlation. And if you supplement with taurine, and this was the thing I think was compelling about this particular study was it was done across a range of animal models. So nematode worms, C. elegans, fruit flies, and mice, and even a little bit of non-human primate data. If you supplement aging or aged animals with taurine, at least up to mice, you can increase lifespan and a variety of health span metrics, and then some evidence for health span improvements in non-human primates. So then the question becomes, is that sufficient evidence that we should start recommending people take taurine supplements? And that, I think gets to the
Well,
bigger
Matt,
challenge.
this data seems actually stronger than what you've been talking about with NAD boosters, though. I
Well,
mean,
I would say it's
not,
stronger, although I
yeah,
would also say it's more limited. So the
yeah,
more studies that have been done on a topic, the more opportunities for failure to replicate or for,
yeah, sure,
so
sure.
I think we have to take it in that context, right?
Right.
Um, but I think that this gets to a bigger question, which is at what level of evidence does the risk reward shift towards reward? And obviously that's also impacted by the risk of whatever supplement it is that you're taking. And
Yeah.
people are going to have different opinions on this. So I said I was a co-author on that study. I don't supplement taurine. So again, take it for what take it for what it's worth.
Where,
I tend to be relatively I tend to have a relatively high bar for adding something new to my quote unquote stack, although it's a tiny stack.
yeah,
There's two reasons. One is we have no understanding of interactions, right? We have no way of predicting how different supplements or drugs are going to interact with each other. So I want to be pretty sure there's a good rationale for anything I add. And number two, while I'm not somebody who is super worried about impurities in supplements and supplement manufacturing, it is the case. There are impurities in supplements, and the more supplements you're taking, the higher levels of those impurities that are going to get in your body. And as we're learning, we're constantly being exposed to all sorts of stuff in our environment
yeah,
that probably isn't good for us. So I don't want to unnecessarily add to that. And much, like we were talking about with the biological age tests, the supplement industry is a freaking
yeah,
mess,
yeah.
right?
It's a mess,
And
and it's
so I just don't trust supplement companies writ large. Again, I think they're good
yeah.
people. There are companies that are trying to do it right, but as a whole, you need to be very cautious.
If there's anything out there that really gets you excited about things that maybe we're not that we don't know that much about yet, maybe they're fairly early.
Yeah.
What kinds of things are out there that you're looking at saying, "Wow, that's really exciting."
Yeah. Well, so let me start with something that I don't think it's not new for sure. But I think there are some things that maybe still, even though people have heard about it, still aren't being emphasized enough, right? So I think a lot now about, preventative diagnostics and which work and which don't work, and things like MRI. I actually just got a clearly
Yeah,
scan, heart scan. I think and I'm not saying everybody should get either of those. That's a longer conversation, but I think there are lots of things that people can do to find things early and take steps to prevent disease before it happens. I would put hormones in that bucket. I think even though there's a lot of talk about hormones, there's a lot of irresponsible hormone stuff going on out there, but a lot of people could benefit from, if your hormones are dysregulated, getting that
yeah,
fixed, men and women.
yeah,
Different conversations, again, longer
yeah, yeah,
conversations. So I think there's a lot to be done there that can have a huge impact on people's lives. Now, if we get to the sort of, geroscience, longevity, pure longevity science world, we touched on epigenetic reprogramming. I'm still waiting to see, is that going to be a real game changer or not? Super interesting technology. The idea is you can sort of use these Yamanaka factors
right.
or Yamanaka factor light to partially reprogram, not all the way back to a pluripotent state, but to reset that age-related
Yeah.
epigenetic damage.
No, that there was stuff through cocktails on that as well, right?
Mhm.
Not
Yeah.
just genetic reprogramming necessarily.
People are working on chemical
Yeah.
cocktails that can do the same thing. None of them, at least the data I've seen, are anywhere near as
Uh-huh.
effective. But at least in theory, yeah, there are different ways you can accomplish epigenetic
Yeah.
reprogramming. maybe through small molecules, maybe through other strategies that don't require gene therapy or transient gene expression. Yes. And so it's an open question. Is that going to be as effective as rapamycin? Is it going to be more effective as rapamycin? Is it going to make old people young again? I don't think it's going to do the latter, but I think there's an opportunity. It could be more effective than rapamycin. So far, nobody's shown that, but I'm intrigued. I would like to see. You mentioned senescence very briefly. I think the idea of senolytics that can clear senescent cells is intriguing. There's debate about how we define senescence, but I personally think it's pretty clear. There are cells that accumulate in our bodies with age that give off inflammatory signals. Whether we call them senescent or not, I don't care. But can you find molecules that clear those cells? I think that's partly how rapamycin works, is it blunts the inflammatory signaling and then allows our immune system to clear those cells. I think immunotherapy against age-related damage or against senescent cells, very intriguing. So those I would all put in the bucket of technologies that are close, and we'll know in the relatively near future, the next 5 years, how hard it's going to be to move these things into clinic and maybe what the effect size is going to look like. Last thing I'll say is in this space, those same technologies and things like rapamycin, I think the opportunity to have significant impacts on health span and lifespan in companion animals, people's pets, cats and dogs, and maybe other companion animals, I'm really excited about, because I think that will happen. Like there's no barrier to that happening other than, will and resources of actually doing it. I think that's a big deal. Again, it's not human longevity, but so many of us love our companion animals. They're part of our family. We can give a family member 20, 30% more healthy life. That's a big deal. And I'm pretty excited about that.
Matt, you mentioned you had your own podcast now. I actually did not know that. So tell us a little
So
bit
it's called the Optispan podcast. Optispan stands for optimal health span. Easy to find. Check it out. All sorts of stuff in there. Some of it's provocative. Some of it's, biology of aging straight up.
Well, I could continue asking you questions all day because I'm completely fascinated by this stuff. I really do appreciate you being on the show today.
Sure thing. My pleasure.
Thanks for listening. A quick reminder that while I am, in fact, a surgeon, nothing I say should be construed as medical advice. Now, make sure to include your physician in any medical decisions you make. And also, if you're enjoying the show, please make sure to show your support with a like, share, or subscribe.